Welcome to this tutorial video. So in today's lesson, we'll look at what we call cholesterol metabolism. Cholesterol metabolism. So this is a topic that you come across in biochemistry, especially those that are in different universities and are doing health-related programs. It's very important for you to understand how the body metabolizes cholesterol. Now, before we even go further, I would like to state that I know that most of you may have come across a component in science which talks about human beings obtaining cholesterol from the diet. Okay, for example, from food sources, maybe from butter, maybe from cheese, maybe obtaining cholesterol from such kind of foods. Now, we will not really focus on that. We will not focus on how the human body can obtain cholesterol from food, but we'll focus on how the human body can naturally, or how the human body can synthesize cholesterol on its own. And that's what we basically call cholesterol synthesis. Now, coming to this term, metabolism, this is something that, of course, students who are in health, programs usually understand. When we say metabolism, we simply mean that it's either a process that is going to inform, that's going to be about formation of something or breaking down of something. But in this particular video, we'll primarily look at how cholesterol can be synthesized, specifically cholesterol synthesis. Now, you must be wondering to say, but why is it important for us to understand how cholesterol is synthesized? Well, this cholesterol plays a lot of physiological functions in the body. For example, it is used in the formation of vitamin D. Apart from that, maybe you've heard somewhere to say this cholesterol is used in the formation of hormones, okay? Specifically, the steroid hormones. You find that this cholesterol molecule is a precursor molecule that is used in the formation of those particular, hormones. Apart from that, you may have heard that cholesterol is also part of the plasma membrane, the cell membrane. So basically, it's very important, for somebody that is studying health programs or that are related to those particular programs to understand how this cholesterol is synthesized. So that's what we're going to look at in this video. Make sure that you subscribe to this YouTube channel so that you have access to more upcoming videos, because this will just be part one of cholesterol synthesis. Once you subscribe, you will be notified to say another video on this particular topic has been uploaded. Alright, so let's start. So basically, I'm showing you this picture so that you just appreciate the concept of cholesterol synthesis. And if at all there were abnormalities that were to take place, then somebody would have what we call cholesterol plaque. So you can see that you've got this artery. Of course, we all understand that an artery is just a blood vessel which transports blood. Now, imagine a situation whereby you've got a cholesterol plaque. What we mean a cholesterol plaque is the collection or the assembly of cholesterol at one particular point in the blood vessel. But you can see that this cholesterol plaque is more like blocking the flow of blood in this blood vessel. So you don't want that situation, because this can bring about hypertension. It can bring about what we call myocardial infarction. If at all there is blockage of blood supply, or what we call heart attack, it can come about. So you don't want this situation whereby somebody is going to be having this cholesterol plaque in the blood. You can even see there on this particular end, you can see that there is this blood that is being pumped from the heart, but this cholesterol is kind of blocking this, blood supply. So it's very important for you to understand how this cholesterol is metabolized and how it can affect certain systems in the body. But for now, let's make progress and see how this cholesterol can be synthesized. All right. Now, these are just some food sources, of course, that contain cholesterol. For example, the hamburgers, the fried chicken, the French fries, the real meat, the egg yolks, the ice cream stuffs. So these are just basic foods that we can eat as human beings, and from these food stuffs we can obtain cholesterol. Of course, via the GIT, they're going to be digested, then we can obtain cholesterol from them. Now, if you remember very well, in my introduction, I mentioned that we will not really focus on these. What we want to focus on is, what if somebody is not eating these hamburgers? What if somebody is not eating these fried chickens, these egg yolks, or this ice cream that contains cholesterol? we want to find out or understand how the body now can naturally synthesize this cholesterol on its own. And in this video, we will definitely look at that. All right. Now, the other thing that you must know, okay, is the primary functions of this cholesterol. I think I did make mention maybe of some functions in my introduction. So the essential functions of cholesterol, because remember when you're looking at any particular biochemical pathway, it is very important for you to understand its significance, because why must you learn about it? So it's important for you to understand what cholesterol does in the human body. I think I did mention to say this cholesterol can be used to synthesize what we call vitamin D. So we need vitamin D because it helps in bone formation and everything. So this cholesterol acts as a precursor molecule for formation of what we call vitamin D. Apart from that, you can see that this cholesterol is also part of the cell membrane. Okay, it does what we call maintaining fluidity of the cell membrane. So this cholesterol is part of what? The cell membrane. And we know that this cell membrane does a lot for the cell. Apart from that, this cholesterol is also part of what we call nerve insulation. Okay, nerve insulation. So it's also a molecule that is used in or that is part of the structures of the nerve, and we know what the nerves do. All right, transmitting those nerve impulses, those electrical signals, and the stuffs. So that's basically what also cholesterol does. Apart from that, we can see that this cholesterol is used in the production of hormones. Very, very important. Because as human beings, we are under the control of hormones that we cannot decline, because we've got what we call the endocrine system, and they are what we call steroid hormones. And those steroid hormones, what helps in making them is cholesterol. So it's very important for you to understand how this cholesterol is synthesized. Apart from that, we've got what we call bioproduction. So in secondary schools, I believe we were almost all of us that passed through secondary school, we were taught that bio is basically, some form of acid that is used to emulsify fats, okay, or to make fats soluble, okay, or to digest fats in short. So this particular cholesterol, as we make progress, you understand that it is also part and parcel of the formation of that particular bio. So that's what cholesterol does. Summary, we've said it is used in vitamin D formation, it is part of the cell membrane, it is used in the production of hormones, it is used, or it's among the component or the structures of a nerve. Apart from that, this cholesterol is used in the production of bile. So very important points there. Now, just key definitions before we even go into the actual details of what this cholesterol synthesis is all about. So you must understand what we mean when we say cholesterol synthesis. So I must make mention that many of the times you come across maybe books or tutorial videos on YouTube that are going to use the term de novo synthesis of cholesterol. Now, when we say de novo synthesis, it's a term that describes a biochemical pathway that involves formation of a molecule from scratch. So that ability of the body to form a molecule from scratch, we call it de novo synthesis. This term may not just be used in cholesterol, but they can even say de novo synthesis of what? Fatty acids. They're just telling you that this is the process that is going to involve the formation of what? Fatty acids from cholesterol. So in this particular instance, we're saying that cholesterol de novo synthesis. So basically what we're saying is that this is a process that refers to the process by which cells create cholesterol from simpler molecules such as what? Acetyl-CoA. So if you've been on my YouTube channel, acetyl-CoA is a molecule that you are familiar with by now. So basically, cholesterol de novo synthesis is a process that involves the formation of what? Cholesterol from acetyl-CoA or acetyl-CoA. So that's what we're going to do. Now, I know that some of you are already familiar to say, cholesterol is a complex molecule. It has got about 27 carbon atoms. But this acetyl-CoA that we're talking about, it is only a 2-carbon molecule. Yes. No wonder we're calling it de novo synthesis of cholesterol, because from this small cholesterol, from scratch, we will be able to synthesize this 27 carbon molecule, which is cholesterol. And that's why I've defined it as cholesterol de novo synthesis is a process that involves the formation of cholesterol from acetyl-CoA. Now, it is written there to say this cholesterol, it is an amphipathic lipid. Amphipathic, it simply means that it has got the soluble part and the part which is not soluble, what we call hydrophilic part, which is soluble, and then the hydrophobic part, which is doesn't dissolve in water because it's hydrophobic. So that's what we mean when we say it is an amphipathic lipid. Apart from that, take it from me at this particular point, this cholesterol that we're talking about contains 27 carbon atoms. Imagine we're going to move from acetyl-CoA, a 2-carbon molecule, to form this 27 carbon molecule. It's a very simple process, and I'll make sure that everything is summarized, everything is well understood. Okay? Apart from that, vitamin D3 is formed from what you call 7-dehydrocholesterol. Now, in my previous slide, I showed you to say this cholesterol acts as a precursor molecule for formation of what? Vitamin D. So that precursor molecule that forms vitamin D is what we call 7-dehydrocholesterol. Of course, it's beneath, it's under the skin there, because I believe we've always been taught to say if at all we are to find ourselves on the sun, especially in the early hours, like from 9 to 10 hours there, we can obtain vitamin D from there. What it simply means that is when the sun is, acting on us, especially on the skin, okay? If you are receiving those right rays, it's able to convert this, 7-dehydrocholesterol into what we call vitamin D. All right. Now, you must also understand that basically 1 gram of cholesterol is produced per day on average. 1 gram of cholesterol is produced per day on average. Now, I'm not talking about this cholesterol that we are going to eat from food substances. No, I'm talking about cholesterol being synthesized from the body in a natural way, in what we call cholesterol de novo synthesis, which we've defined as forming cholesterol from acetyl-CoA. So 1 gram of cholesterol is synthesized in the human body on average or per day, 1 gram of cholesterol. Now, cholesterol has got a parent nucleus. A parent nucleus. So when you say parent nucleus, I believe that some of you have already come across this cholesterol structure. We've seen those ring-like structure that it has. We call them the parent nucleus, which we refer to as the sterin. Okay? Which refer to as what? The sterin. Some of you can, if you don't want to stay sterin, you can use this long term that I've put for you down. Maybe you've heard of it. This term that is starting like cyclopentan or something. So you can use this particular term if you don't want to stay sterin. But in case you come across it, it's just describing those four to five, okay, nuclear structures, cyclic structure that we see on the cholesterol molecule, those kind of boxes that have got those carbon atoms. I'm going to show you very soon. So that is what I needed you to know on this particular slide. All right. Now, biosynthesis of cholesterol. So remember, in all my videos, I've always advised you that it is very important for you to understand when a biochemical pathway is taking place in the body, because you'll be dealing with patients. You must understand to say if a patient is in this particular state, are they undergoing cholesterol synthesis or not? Are they undergoing glycolysis or not? So this is why I always emphasize in my videos that a student must understand when a biochemical pathway such as cholesterol synthesis is taking place, such as fat acid synthesis is taking place. When is it taking place? So what are we saying? Biosynthesis of cholesterol, or what we call de novo synthesis of cholesterol, takes place when one is in a well-fed state. In a well-fed state. So for our body to be able to synthesize this cholesterol from scratch, from that acetyl-CoA, we must be in a well-fed state, meaning we must be eating well. There shouldn't be a situation whereby we're starving and we're expecting cholesterol synthesis. No. If we're starving, we're fasting, we're not eating, there will be no way the body will be synthesizing this cholesterol. There will be other pathways that we've covered already. So somebody who is in a well-fed state will be synthesizing cholesterol. Apart from that, having excess of glucose. Okay, having excess glucose. So of course, when somebody is having excess glucose, meaning they'll be having enough energy. So that excess glucose, now you understand, as we make progress, it is the one that will bring about formation of what? Acetyl-CoA, which is our precursor molecule for formation of what? Cholesterol. Okay. Now another part is that this process is promoted by what? Insulin. Remember that whenever the blood glucose levels are high, and in this particular case, we've already said that when somebody is having high glucose levels, or the glucose levels are in excess, of course the body is going to produce that insulin, of course by the help of the pancreas. Whenever the blood glucose levels are high, then this insulin is going to promote what? Glycolysis. From that glycolysis, we're going to obtain pyruvate. From pyruvate, under aerobic conditions, there will be formation of what we call acetyl-CoA. This is now when this acetyl-CoA is going to become our precursor molecule for formation of this cholesterol. And in the other way, or however, this cholesterol synthesis is inhibited by what? Glucagon. Because remember that glucagon is always released when the blood glucose levels are low. So in this particular case, we've said one is in off-fed state, meaning the blood glucose levels are high. One is having excess glucose, meaning their blood glucose levels are high. So you don't want to produce glucagon. So glucagon can't or can never, support this process. Things like, or neurotransmitters like epinephrine, we would definitely not promote this process, because epinephrine, being a neurotransmitter or a hormone, it promotes the formation of glucose instead of breaking it down. So it can, support this process. And last but not the least, they are what we call statin drugs, or what we call lipido drugs. So these drugs are used in inhibiting what we call cholesterol synthesis. Okay, there are patients who have got what we call hypercholesteremia, or they've got too much cholesterol in their blood. You don't want a situation whereby they'll keep on forming that cholesterol. And then that cholesterol you saw on my second slide as I introduced this topic, why it was blocking, the supply of blood. So we give them what to call statin drugs to prevent the formation of this cholesterol, because you don't want it too much, because it can bring about something like heart attack, myocardial infarctions, okay, ischemic, kind of conditions, and you don't want that. So we're going to give them the statin drugs. There are specific examples in my notes. I'm going to show you those. Okay. Now, apart from knowing when this process takes place, it is very important for you also to understand the organs that are involved in this cholesterol synthesis. Now, the liver. I know that most of you know that I've abused the liver in most of my videos. If I talk about amino acids metabolism, I involve the liver. Gluconeogenesis, I involve the liver. So it's just reminding you as a medical personnel, as a student who is studying these medical related courses, that the liver does a lot. The liver does a lot. So again, this cholesterol synthesis, 80% of it takes place in the liver. Of course, 10% of it will take place in the small intestines. Then about 5%, sometimes it may take place in the skin. Okay? Apart from that, maybe the adrenal cortex, because in the adrenal cortex, those that have done the endocrine system, they will understand that we've got what we call steroid hormones there. We've got the androgens, we've got the cortisol hormone, the aldosterone hormones. All those come about, or their precursor molecule is cholesterol for them to be formed. So this is why you find that it can take place there. Apart from that, the ovaries, the testes, the placenta, all these produce sex hormones. Okay, so these sex hormones, they are steroid hormones, and I said in my introduction that steroid hormones, their precursor molecule is what? Cholesterol. So this is why that you find that these organs are involved in cholesterol synthesis. Now, take note that cholesterol synthesis takes place, if I'm talking about the primary organelle involved, it takes place both in the cytoplasm and the endoplasmic reticulum. The cytoplasm and the endoplasmic reticulum. So look at how everything happens. So that is what I needed you to know on this slide. Summary on this slide, we've said that when does cholesterol synthesis take place? When one is in a well-fed state, when one is having excess blood glucose levels. Okay? Or when I say excess blood glucose levels, it means that the blood glucose levels in the blood are high. Okay? And then we've said this process will be promoted by insulin. And then things like glucagon hormone, epinephrine neurotransmitter, statin drugs are going to inhibit this process. The organs involved, we've said here that these organs are the liver, the intestines, the skin, adrenal cortex, ovaries, testes, and the placenta. Okay, because this placenta, of course, we understand that when a woman is pregnant, they produce what we call progesterone hormone that makes the environment for the uterus conducive. So this is why you find that this progesterone hormone, of course, it's also a steroid hormone. Okay, this is why I find that the placenta can also, be part of what you call cholesterol synthesis. Now, those that were wondering to say, he has been talking about cholesterol. He has been talking about cholesterol. This is the general structure of cholesterol. Okay, this is the general structure of what? Cholesterol. You can see that it has got these rings. We number them like A, B, C, D. And these rings are what we call the sterin, or the parent nucleus that I talked about. Okay, so interestingly, you can see that this cholesterol, we've got what we call acetate there. That's basically trying to describe acetyl-CoA. So from acetyl-CoA A, we are going to move from a 2-carbon molecule, which is acetyl-CoA A. You can see that we've got a carbon there and a carbon there, just 2 of them. And then we're going to synthesize this 27-carbon molecule. So this is how cholesterol looks like. You can see there that we're starting from the first carbon there. Okay, let me just show you. Okay, we are starting from the first carbon there, and then we are going to end up with 27 carbon atoms. So we are going to go into the actual steps that involve this cholesterol synthesis. Don't worry, they are summarized. So that's what I needed you to know there now. Requirements. In the definition, we already said that we will need our precursor molecule, which is what? Acetyl-CoA. So this is going to be our substrate for what? Cholesterol synthesis. Of course, we are saying that it's obtained from excess glucose catabolism through glycolysis. So you'll be wondering to say, but why is he saying acetyl-CoA? So remember that from excess glucose, that excess glucose will be undergoing glycolysis. If it undergoes glycolysis, it is producing what we call pyruvate. Pyruvate in the presence, as long as we are breathing, we are under what we call aerobic condition, aerobic respiration. That pyruvate is going to form acetyl-CoA. Remember the Krebs cycle? Now that acetyl-CoA, now if it is too much, or it is in excess, if it is in excess, will be used in the formation of what? Cholesterol. Of course, there will be enzymes that we're going to need. We're going to talk about them. Apart from that, we're going to need about 16 NADPH molecules plus hydrogen. Okay, these we can obtain them through what we call the HMP shunt. Okay, those that have been following me, they know that the HMP shunt produce what we call, NADPH. Okay, the HMP shunt, the hexose monophosphate pathway shunt, or what we call the pentose phosphate pathway shunt. I explained how this comes about. So we're going to need NADPH, and we're going to need about 36 ATP molecules to be able to synthesize that cholesterol. So it's an energy requiring, kind of process. All right. Now, it's very important for you to understand what is going on this diagram before we even start talking about the actual steps that are involved in cholesterol synthesis. Now, looking at the biosynthesis of cholesterol, for now, just ignore these particular, diagrams like the liver, the intestines, because these are just trying to remind you of what I talked about to say the organs involved in cholesterol synthesis. I said we've got the liver, the intestines, the adrenal cortex, the testes, the ovaries. I talked about this, the skin. Now, here is what I want you to understand. Remember, I told you that when we are talking about cholesterol synthesis, we are not talking about cholesterol that we are obtaining from the diet. No. We are talking about cholesterol that will be synthesized by the body from scratch. And we've said that it will primarily takes place. It primarily takes place where? In the liver. Now, what I want you to get from now is that whenever somebody has got high blood glucose levels, or when the blood glucose levels are high, they are the ones that can bring about cholesterol synthesis. I know you're wondering to say, but glucose maybe is coming from carbohydrates. Yes, it's coming from carbohydrates, but remember the precursor molecule is acetyl-CoA, and these carbohydrates can bring about what? Formation of acetyl-CoA. How? Let me explain. So let's take, for example, there. Let me just zoom in there. Let's say you've got this person who has been eating rice, potatoes, shima almost the entire day. They have got high blood glucose levels, and then the body's like, this person has not eaten anything to do with cholesterol, and I need this cholesterol for what? For vitamin D synthesis. I need this cholesterol for production of hormones. So what do I do with this shima, or what do I do with this rice? Okay, so the body knows that, okay, from this shima, from this rice, from these sweet potatoes, there's glucose. So this glucose, if it's in excess, it's going to undergo glycolysis that is going to form what? Pyruvate. Now we all understand that pyruvate can form acetyl-CoA under aerobic conditions in the presence of oxygen. So if there's too much glucose, too much pyruvate, too much pyruvate, too much acetyl-CoA production. Now, this acetyl-CoA is in the mitochondria. If you're looking at my screen right now, the box that you are seeing there is describing the mitochondria, as you can see. Then the outside part is what? The cytosol. The cytosol or the cytoplasm, which is going to involve this cholesterol synthesis. And also including the smooth endoplasmic reticulum. Now, before we even come outside there, let's just go in a bit inside. So if you've been following me very well, you understand that cholesterol will not leave the mitochondria. Acetyl-CoA will not leave the mitochondria as acetyl-CoA. It's not possible. Why? Because human beings do not have a transporter on this membrane of this mitochondria that can transport this acetyl CoA from the mitochondrial matrix outside into the cytoplasm. So what does it mean? Sir, you said that cholesterol is what we're going to need. I mean, acetyl CoA is what we're going to need to be able to form this cholesterol. And we're saying here that this acetyl CoA can't leave the mitochondria. So it leaves via what we call the citrate shuttle. What do we mean the citrate shuttle? Okay, let's just do a bit of the Krebs cycle so that we understand what is going on. So let's take, for example, we start talking about the Krebs cycle. We know that if this pyruvate under aerobic conditions is going to form acetyl-CoA. Acetyl-CoA in the Krebs cycle, specifically in the mitochondria, it's going to combine with what we call oxaloacetate. Okay, so this oxaloacetate combined with acetyl-CoA, they're going to form what we call citrate. Okay, citrate, I think we are all familiar with it because this is just a 6-carbon molecule, because acetyl-CoA is a 2-carbon molecule, oxaloacetate is a 4-carbon molecule. When they are combined, they form what we call citrate. So this citrate now, since acetyl-CoA can't leave the mitochondria, so the body has got a way to do it. So if it knows that if acetyl-CoA can't leave this mitochondria, but I need it for cholesterol synthesis, so what do? I'm going to combine it with I'm going to combine acetyl-CoA plus oxaloacetate, then I form citrate. Now, this citrate is able to leave the mitochondria. So, this citrate is going to exit, or it's going to be shuttled out of this particular mitochondria via what we call the citrate shuttle. Then when this particular citrate leaves the mitochondria now, the body is like, okay, what did I use to form citrate? I used acetyl-CoA and oxaloacetate. So it simply means that this citrate, inside of it contains acetyl-CoA. Then outside now, it's going to break it down into what we call acetyl-CoA and oxaloacetate. You've seen how it has done it. It has used a different method of, getting this cholesterol into, this acetyl-CoA into the cytoplasm. So this citrate now will be broken down into what we call acetyl-CoA and oxaloacetate. And then you can see that all carbons of this particular cholesterol that we're going to get will come from this acetyl-CoA. Then this acetyl-CoA now can be used to form cholesterol. So what I'm just from describing is what we call the citrate shuttle. Now don't worry, I know that you can see on top there's the NADPH, there's the malic enzyme, the HMP shunt. What is just trying what these are just trying to describe here is that when you're forming this cholesterol, you just don't need acetyl-CoA. You need the NADPH, nicotinamide adenine dinucleotide phosphate plus hydrogen is the abbreviation for, is what we mean when I say NADPH. Now, this NADPH can come from what we call the HMP shunt. The HMP shunt. Then there's what we call cytosolic isocitrate dehydrogenase. Okay? On this particular pathway as well, you can obtain what we call NADPH. Then there's what we call the malic enzyme pathway, which can also bring about formation of this NADPH. We need this NADPH, okay, as a reducing agent as we synthesize this particular cholesterol. So next time as you watch part 2, okay, make sure that you subscribe to this YouTube channel, because if you don't subscribe, then you may not have access to the second part of this particular cholesterol synthesis mechanism. Now, the other part that I want you to get is that I said that the liver is the primary organ for cholesterol synthesis. Okay, we're saying that the liver plays a central role in the regulation of body's cholesterol hemostasis. Of course, hemostasis simply means the balancing of cholesterol, maintaining the internal environment, okay, maintaining the internal equilibrium of cholesterol inside the human being. So, what is this picture trying to depict or trying to tell us? We've said that cholesterol synthesis is a process of forming cholesterol from acetyl-CoA. And then we've talked about how acetyl-CoA can be shuttled from this particular mitochondria into the cytoplasm. Now, if at all we're talking about the liver, so the liver has got there are different ways in which the liver can obtain this particular cholesterol. For example, one way is what you go through cholesterol being collected from what you call extrahepatic tissues. In science, when we say extra, it simply means outside. So it simply means outside the liver tissues, because the hepatic tissue is talking about liver tissue. So there's what we call HDL, high density lipoproteins. Okay, these are the ones that collect excess cholesterol, let's say your kidney is not using this cholesterol, then this HDL is going to get to say, okay, this cholesterol is not doing anything here. Let me take it to the liver. That is one way the liver can obtain cholesterol. Apart from that, the liver can obtain cholesterol from what? From that. You can see butter, milk, egg yolk. These can be digested and they form what we call chylomicrons. Then these can be used to form cholesterol. They can bring about cholesterol. So you can see there's what we call cholesterol pool in the liver because the liver is getting cholesterol from the HDL. It's getting cholesterol from what? From the diet as well. Now, the liver can also synthesize cholesterol on its own via what we call de novo synthesis of cholesterol in the liver. That is the third part. So 3 ways in which the liver can obtain cholesterol, on which this particular cholesterol pool can be increased. Now, what can this, because remember we said that the liver must maintain what we call homeostasis. You just don't want a situation whereby you're just forming cholesterol, then it's not being used. The liver is going to be full of cholesterol. So you don't want that situation. So the liver, what it's going to do is that this cholesterol that it's getting now from the HDL, okay, from the diet here, and also from the novel synthesis, from acetyl A, as we are forming cholesterol, it can use that cholesterol to form what we call bile acids and bile salts. I'll talk about the difference between the two because there's a difference between bile acid and bile salts. Okay? And then this cholesterol can also be excreted in form of what? Bile. Apart from that, it can be secreted in what we call VLDL, very low density lipoproteins. So this is how the liver balances or maintains homeostasis of cholesterol in the body. It's getting cholesterol from the HDL, from the de novo synthesis, which we're going to talk about, then the diet, and then it's using it for formation of bile acids, bile salts, okay? And then also formation of what we call VLDL. So next time, we are going to look at part 2 of cholesterol synthesis so that we can look at the actual pathways, the enzymes, and some sort of diagrams that are involved in cholesterol synthesis. So make sure to subscribe to this YouTube channel so that you have access to the next video. Thank you.